Yeah. There we go. All right. Good evening, everyone. I'm Connie Lee. I'm CEO of Angioma Alliance, and I am pleased to be joined this evening by Dr. Issam Awad, chair of the Angioma Alliance Scientific Advisory Board, and he's got a lot of other things that I'm going to say about him. He is the University of Chicago's John Harper Seeley Professor of Surgery. He leads the Neurovascular Surgery program and co-leads the Stroke and Neurovascular Care team. And he's the director of our CCM Center of Excellence, our flagship CCM Center of Excellence at the University of Chicago. Also here with us is Agnieszka Stadnik, and she is working with Dr. Awad, and I will let her introduce herself when it's her turn, or let Dr. Awad introduce her. Before we really get rolling though, two things. One, if you have a question, please ask it using the Q&A, and you'll see that at the bottom of your screen. Click it, ask the questions there. It's easier for me to keep track of them. And also, immediately following this webinar, there will be a support group session, and we'll be putting the link to the support group in the chat toward the end. And you can just click it and that should get you in. So welcome, Dr. Awad.
Thank you. Thank you so much, Connie. And Agnieszka actually is the manager of our clinical research operation. And I think many of you who have come to Chicago at one point or another probably met her, as she made you sign papers for consenting for various things. We are running many research projects. How many are active right now, Agnieszka, with the IRB?
We have 8 or 9 active currently.
8 or 9 active ones, with 3 big clinical projects funded by the NIH that have to do with hemorrhage and cavernous angioma, in addition to our lab work. So I think we've been very busy, but the goal today is to really give you a little update on what is happening with the Atorvastatin trial, as we're coming close toward the last stride in completing enrollments. And I'll give you a very brief update on what is happening with the trial readiness project, which is similar to the Atorvastatin trial, but is occurring at San Francisco, Utah, Mayo Clinic, etc., where drug is not given, but you follow with those teams and get the same imaging. And we'll tell you what we're learning from that project. And also the CASH biomarkers project will give you a little update on that. So for the Atorvastatin trial, just a brief summary. This is based on the fact that Atorvastatin at a high dose in humans, at a dose of 40 to 80 milligrams per day, which is a very commonly used dose for secondary stroke and heart attack prevention. That same dose of Atorvastatin, or Lipitor, actually inhibits one of the main signaling pathways called Rho kinase that drives cavernous angiomas in part, among other signaling pathways. So because of that, we had worked for nearly, I was just thinking a little bit about when we started looking at that possible drug use, and it's been actually 10 years. So when we first put the drug in mouse and saw an effect, and then we had to do a lot of work on dosing and comparing different statins and so forth before we received the grant from the NIH to do this trial, which we are coming toward the second. We are in the second half of the 4th year of the trial. The trial is planned to last 5 years, and I will show you for a second. Agnieszka, do you want to go ahead and share your screen? And I'll show you how our recruitment has taken place. So what has happened with the trial is, if you notice, from point 0 till 12, 2018 or November 2018. So we received the grant in July and then enrolled the very first patient around November. And that patient, of course, completed the trial after 2 years. But as we continue the enrollment, what happened is the COVID first wave and the second wave that caused the pausing of the projected enrollments, both for the first pandemic and the second pandemic. Now, we were able to continue to work during that time, and we enrolled actually a few patients during that time that were either in Chicago or were able to come. So we never totally were shut down, but it was substantially delayed. So if you add those two periods, basically that added a very, very significant time to the trial. We should have actually probably finished enrollment around now if it weren't for the COVID and for the initial NIH delay. As it is, we are now at, I think, 63 or enrolled. Is that correct? 63 enrolled, and we have actually two very tentative enrollments this coming week. So if we actually enroll them, we will be at 65. And the trial is supposed to enroll 80 patients. So we think we will complete the enrollment during this current calendar year. And then the patients, the last one, still has to be followed for 2 years before we can have the results. So the more we enroll sooner, the more we finish sooner, and that's how trials go. And in reference to other trials during the COVID era that are not investigating COVID itself, but really studying other things, we received a lot of commendations by the NIH for our continuation and activity with this trial. We are actually under budget in the sense that the money that was delayed in the enrollment will be used to complete the enrollments. So we have basically money in the bank to complete the trial through the NIH. And I think what we need is an additional 12+ patients to step up who have hemorrhaged in the prior year and whose lesion had not been resected. So this remains the requirement. So you have to have had a new symptomatic hemorrhage, symptoms plus blood on MRI. And then in the past year, which is very good, you know, so if you bled 6 months ago, 7 months ago, you still have a number of months to enroll. And then once you enroll, you get followed for 2 years. There are 3 visits required to Chicago at enrollment, at 1 year and at 2 years. And you receive a $500 stipend for each of those visits if you are coming in from outside Chicago, and a much smaller stipend if you are in the city. And the idea here is to help a little bit with the flight and travel. We know it doesn't cover it all, but it does. And of course, all the drug and the MRIs, the very special MRIs that are used in that trial, are all covered by the NIH, including consultation with a statin expert about any symptoms you might have during the trial. So that's the progress of enrollments. There were several milestones in the trial that were completed this year. There are regular prescribed safety reviews, and the safety reviews have gone extremely well with no, so the safety reviewer who does not work with us, is at another institution, actually gets to see who is on drug and who is on placebo. And even for her, we call them A and B. We don't tell her which is which. And if there is a difference that is worrisome about A or B, then she is required to report to us and to the NIH. And in fact, the safety reviews have been very, very good. The symptoms on statin have been also very, very low. Most patients, I think our compliance among the enrolled patients are in the high 90%, and I think people are really being good troopers about the trial. There is an app now that helps you interact with us from the very beginning till the end of the trial. There is also another major milestone that happened this year, which was what we call the midpoint analysis. So midpoint analysis has happened when actually about almost 3/4 of the patients have been enrolled, but it's called midpoint because half of the follow-up visits were completed. So that half of the total follow-up visits of the trial were completed. And this happened this past fall. And we did the halfway review, both for futility and for recalculation of the sample size. So the futility means if that doesn't look like there is a chance that there is a difference between the placebo and the statin, we didn't need to continue the trial. So there was no futility. So there is some effect there. We don't know if it'll be significant or it'll continue for the rest of the trial, but there is an effect, a difference between the statin and the placebo. And then the second thing is we recalculated the sample size based on the actual measurements in the real patients rather than projections that we had on pilot patients before the trial. And in fact, the numbers came exactly as we had predicted. So many of our assumptions in the trial are bearing out. We had done them with about 15, 17 patients initially, and they stood out with us having completed 80 follow-ups. So it is really very, very good. So I think that's where we are with the Atorvastatin trial, and I'll answer any questions later on that. Let me move straight to the trial readiness. So the trial readiness is very similar to the Atorvastatin trial, except the sites at Mayo Clinic and UCSF and Johns Hopkins and others had not yet perfected doing the biomarkers on the MRI. So part of the aims of the trial readiness was for them to show that they can do them as well and in a precise way as they can be done in Chicago. And so you helped, many of you who followed up in San Francisco or Mayo Clinic and those other sites, helped us with actually showing that the same measures can be made at multiple sites. We also learned, which will be very, very important for all trials in the future, that if you have a certain number of patients that you see per year, what is the percent that can be enrolled in trials based on various entry criteria. So if hemorrhage is the entrance criterion, about 12 to 15% of patients actually are eligible and do enroll. So this is, we learned that, and now everybody has their calculator out, because a lot of people say, well, I see 30 patients, I see 40 patients, but that means for hemorrhage, you'll be able to enroll, 4 or 5 patients a year if you see 50. So that's kind of where we learned about that. The other very important thing is we had never known about changes in the quality of life of patients over time. There have been a lot of studies about how cavernous angiomas impact our quality of life, the effect on anxiety, the effect on function, the effect on depression, and other facets of quality of life have been very well studied. I know Connie also has been very involved in studying that. But what has never been done until the trial readiness project is really look at the quality of life over time in trial eligible subjects. That is important because if we think a drug is going to alter the quality of life, then there has to be a change in the quality of life that can be changed by the drug. So, so we're looking at that, and so that trial Readiness is now in its 5th and final year. And as a result of it, we have gone to the FDA with the MRI biomarker, that is a measure of how much iron there is in the brain. And we are preparing a letter of intent that will certify this biomarker to be allowed to be used by drug companies to certify their drug if they so choose. So it will be an available monitoring biomarker that is endorsed by the NIH. So I think those are all important landmarks that we've done with these 2 projects. And I want to tell you next about the biomarkers project. And those of you who come to Chicago, you know that when you go into the MRI machine and they put the IV, usually Agnieszka or someone else from the team is there to ask you if you are okay with us obtaining a blood sample. I think Justine just joined us. Justine, you can turn things on if you want, so I introduce you.
Sorry, I was having some technical difficulties.
No problem. We are glad. So Justine is our research nurse, and she has been just nothing short of phenomenal. How long have you been with us, Justine? About 8 months, 7 months?
So June, yeah, I think about 8 months.
Yeah, about 8 months. And I just don't know how our program would be like without her. She's been really amazing, and she's so attentive to detail. And I think anyone who interacted with her by answering the @cash@uchospitals.edu know that she's right there. I know she's there because she gets me on the weekend and Saturday night, and every time she's answering the questions of everybody. So thank you, Justine, for joining us, and it's good to put the name with the face. So the biomarker project is basically aimed to look at 3 questions. The first one is, can the blood tell us whether our lesion has bled in the past year? And as you know, many of you are confused by this. You know, one doctor tells you you bled, another tells you you didn't. Maybe you have an MRI 3 months after your symptoms and the blood is gone and there is old blood, but you don't know when it happened. The reason it's important to know if the lesion had bled in the prior year, because that same lesion is now 10 times more likely to rebleed. That's what's unique about the so-called cash, is that once a lesion bleeds, it's 10 times more likely to do the same than one that never did. So it is consequential when people tell me I have headaches or I've had a seizure, and their lesion doesn't have fresh blood, but their headache was 6 months ago. To be able to tell them whether their lesion, in fact, had a hemorrhage 6 months ago or not. So we compare patients who had for sure a bleed that we know on MRI in the prior year to those who we know for sure on MRI did not have a bleed in the prior year. They have no symptoms and no blood. And we look at their blood, and we are developing a number of molecules that are combined with each other to help answer that question. And this is a large project. It's about $4.2 million by the National Institutes of Health. And it also includes collection of samples from Mayo Clinic Center of Excellence and from UC San Francisco Center of Excellence, because we want to be sure that the same blood test that works in Chicago works at those other sites. And we also received another grant from the Provost Global Initiatives at the University of Chicago that's allowing us to collect the same samples and run the test in Brazil. So we'll be able to validate whether also in another country the test will work. So the first question is, did my lesion bleed? The second question is, which is the more exciting one, and the one actually we're better able to answer, is will my lesion bleed in the next year, a symptomatic hemorrhage or growth? Meaning, will the lesion grow in the next year or cause me a hemorrhage with symptoms? And that phenomenon in the next year is very reliably predictable by a combination of molecules. And we're working very hard now to make sure that the molecules we are discovering are applicable in both sexes, so in males and females. They're applicable at all age groups, and they're applicable in people who have epilepsy and don't have epilepsy, and in familial and sporadic CASH cases, and in brainstem versus non-brainstem CASH cases. So we raised the bar very high here. We have already a hint that the test works in general, but in order to prove that it works for all of you and therefore be able to develop it with a diagnostics company, it is going to take this type of very rigorous work, which we are in the middle of doing. So we're aiming to collect 800 blood samples, 800 cases with blood samples that we follow backwards and forwards. So they can serve for both prognostic and diagnostic testing. And we are close to having collected about 500 of them. So we are doing very, very well with the collection. And of course, this is only in the second year of this project. And therefore, having the sample doesn't mean we've done all the work. There is tons of work to be done on assays and things like that. So if you ask what type of molecules they are, they're going to be a combination of proteins, which we have first worked with. These are proteins that reflect leakage or blood vessel development or inflammation. And also there are going to be microRNA, which are little snippets of RNA that circulates in our blood that actually reflect the activated genes in the brain. And we do find very reliably microRNAs in people who bled that are not present in people who didn't bleed. So if we combine the microRNA with the protein, we will add the precision of the test. And finally, there is going to be what we call a small molecule or a metabolite. That is one that probably reflects our gut. So whether the microbiome is permissive or protective will be reflected by that one molecule. So if we combine all of those together, we think a test of about 5, I'm holding the team to less than 5 molecules, so it is practical. And then we combine the levels of those molecules. So it sounds like it's complicated, but at the end of the day, it's very simple. It's gonna be a blood test that tells you if your lesion is stable or unstable. And I have yet to meet a cavernous malformation patient that doesn't want that answer when they look at their MRI scan. So I think this is a great need. If scientifically it's validated, it will be widely welcomed by the community. So those are the CASH projects, the cavernous angioma with symptomatic hemorrhage, that my team has been working very hard on. I think I just wanna take another 5 minutes to just tell you, and then we'll go to questions, to just tell you a little bit about some of the updates on the basic science side of the world. You know, all of you have heard about the discovery in Nature last June that identified a mutation that happens in the cavernous malformation lesions that's called PIK3CA activation that seems to drive the active or hot lesions. We don't know that that's the only thing that drives the hot lesions, but we do know that if you have it, the lesion will be much more aggressive. The nice thing about that particular mutation is that it can be inhibited by a drug called rapamycin or relatives of that drug. So before we actually can propose that drug for a trial, we have to kind of show that it works on CCMs, whether they have or do not have the PIK3CA mutation, because we can't get to your lesion to tell you if you have it or not in your lesion. And it does look like in the mice, it has an effect at high dose. And now we are trying to work if the dose of rapamycin can be similar to the one that us human beings can tolerate using without a lot of bad side effects. So that work is ongoing, and I think that could be a promising drug. There is a lot of work going on between us and the University of California, San Diego group on the effect of low oxygen in the blood, which may have implications for people who have sleep apnea and other respiratory problems, and what that does to CCM, and also in people who have clotting tendencies, what that does to their CCM. So those are still at the bench stage, but rapidly will find their way to the clinic. And these microRNAs and molecules that we are discovering in the biomarkers were all generated from the work on the mice, and we are now validating them in you guys, in the human subjects. So it is 6:28, and I think this is the update that I wanted to give you, but I know that you have a million questions on this and maybe other things. So let's see what they are and where the discussion takes us.
Okay, thank you. So yes, we have a bunch of questions in the Q&A, and I'm going to start there first and ask you some of the ones that got mailed in a little bit later. The first question is, why is it thought that a statin would be helpful for cavernoma?
Yeah, so this is what I alluded to. So cavernoma develops because there is a signaling, a molecular switch called Rho kinase that gets activated. And that's what makes the blood vessels leak and become proliferative and bleed. So inhibiting that Rho kinase activity actually stops the growth of cavernous malformations as well as their bleeding. And we showed that in the mice using specific Rho kinase inhibitors that are now not yet available to patients. But statin at the dose of 40 to 80 milligram Lipitor actually affects Rho kinase inhibition in humans. In fact, that's why in part it works on the atherosclerosis effect by protecting from atheroma development because of Rho kinase inhibition, aside from the cholesterol. We all take it to lower cholesterol, but the effect on preventing stroke and preventing heart attack really is beyond the cholesterol lowering effect. Statin has this Rho kinase inhibition effect. So that is the reason we think it works, and we have shown that it works in the mice to decrease bleeding in the dose that we are now giving in the trial. So I hope this helps answer that question.
Okay. And then someone is wondering if we already know what the results are of the people who have completed the 2 years using statin.
Yeah, so unfortunately, we cannot open up the blinding until all the trial results are done, because it's really meaningless to have numbers that have not reached the level that is needed for statistical significance. So if we thought that the answer was sufficiently different now, we would not continue the trial. Okay, so there is somebody looking at this. It's not me because I'm blinded. I don't want to be biased, and it's not you because you're also biased. You know, if you think you're taking something, maybe it's working or not working. So we look to make sure that the difference is, if it was going to be already significant, we would have stopped the trial. If people were bleeding much more on the placebo than on statin or vice versa, we would have stopped the trial. If statin was not safe, we would have stopped the trial. So we're monitoring it. There is a difference. The difference will not reach statistical meaningfulness unless the full power of the trial. That's why you need 80 people to show that difference. So I don't know if that difference will persist in the remainder of the trial or will go away, but we won't know until the whole trial is completed. That's how throwing the dice works. You know, you can't keep thinking that you know what you're gonna get until you do enough throwing of the dice.
Okay, a question about the risk of statin. What about the risk of A1C going up due to high dosage atorvastatin, and therefore can it lead to diabetes?
Yeah, so that actually with Lipitor at 80, unless you are already a diabetic where your control may fluctuate with statin or with other things, some people may have to increase their metformin dose or take a little bit closer look at their diabetes. But the majority of the patients we're enrolling in the trial are not diabetic, and they do not develop, nobody develops diabetes from statin.
What side effects have people in the statin trial experienced so far?
So the majority of the patients ask whether a symptom they may be having, because you know you're taking this pill and then you feel something, and then that's what you contact Justine, and Justine runs it by me and by our statin expert. So our statin expert, by the way, Dr. Sorrentino, is world renowned for, he's treated hundreds of thousands of patients on various doses of statin. He's a cardiologist. So he's the one who advises us on this. In general, the symptoms have been, am I feeling tired because of it? Or is my leg pain that I may have caused by it? And there have been a few patients that thought that and chose to stop. How many, Agnieszka, so far were?
I'm sorry, stop.
Yeah, that stopped because of symptoms. I didn't like the
Oh, I
medicine.
think 3 maybe.
Yeah, 3 or 4. So out of the 63, so it hasn't been very high. Usually we run lab tests when you have those symptoms, because if statin is what's causing the muscle pain, usually we can see the muscle enzymes on the test. And I've been able to get back on the phone and reassure the person that the labs are okay and ask them if they want to continue or not. And most people, except for these 3 or so people, have actually continued on the medication. So the compliance is extremely high. It has been very well tolerated. We're not surprised, because that's kind of what statin trials show. If you want to just read the small print about what can happen with any drug, don't even take any Tylenol, you know.
Yeah.
That's what it is.
Okay. So in regards to the statin therapy, if a patient has high cholesterol and CCM, would the atorvastatin dose of 40 to 80 milligrams be effective to treat both?
Yeah, it would, except if you have high cholesterol that warrants being on statin on its own, we tell you to get on statin. You don't get in the trial. The trial is half people on statin, half people are on placebo. Now, where the issue comes up is a lot of you have cholesterols around like 198. It's right around the edge. And you haven't wanted to take statin before, you've tried diet and so forth, and you're kind of holding things there. So in those cases, yeah, you have 50% chance of getting your cholesterol down with the drug. will tell you to continue your diet because I cannot guarantee you that you're not taking the sugar pill, 50-50 assignment. So if you really need it, like you have heart attack and the stent and high cholesterol, and you're already on it, then we're not gonna stop it. So you're in the trial.
Two more statin questions.
Oh, one more thing on statin I wanted to add. So nobody has had the serious adverse events that people read about. So, people read about liver, about muscle necrosis, about things like that, that are what we would call serious adverse events. They're reported in usually 1 to 2 patients per 1,000. And we have not had anybody like that in the trial.
Okay. Question, is statin beneficial for brainstem lesions as well?
Yeah, so we are about 2/3 of the patients in the EPOCH trial have brainstem lesions, and that's not surprising. That's because brainstem lesions, people don't want to dig inside the brainstem to take it out. If it were easy to take out, you'd maybe take your lesion out rather than be on the trial. But people who have thalamic bleeds, brainstem bleeds, deep bleeds are the majority of the patients in the trial.
Okay. And then when will the statin trial be completed?
Yeah, so there are several stages to the completion. So completion is of enrollment is when we enroll 80. So, this is like you have like when you do fundraising, how close am I to my end? That depends on, if the community embraces it as they have and continue, then we should be done with it in the next few months. If not, it will be delayed. From the point of enrolling the last patient, we have to wait 2 years for the results. So that is the painful thing, but that is what it is.
Yeah.
So the sooner the better, come on down and join it. So we finish it and get you the answer.
Okay. All right.
And this is not just this trial. There are no trials with quick answer. I want to just know now, tell me what I'm on and is it working or not. All the trials are going to be like this and including everyone that is being planned down the pike. Go ahead. Sorry, I interrupted you.
Oh, no worries. So a question about a different trial. I heard that there's a study in Italy with propranolol. Have we seen any results from this study?
Yeah, so the study in Italy is going to be very similar to a few of the trials that are going to be maybe used by some groups, which are looking for signal effect and safety, and not necessarily powered to show a benefit of the drug like the Atorvastatin one is. So they have completed enrollment. They are completing the follow-up of their patients, but they are the first to admit that they do not have enough patients to be able to show a difference between propranolol and the placebo. They are going to see if there is a signal effect. They're also looking at all the biomarkers that came out of Chicago. So they're using them all and hoping that some signal would come up. And based on that, they would then go to what they would call phase 2B trial that is aimed to show an effect.
And then a different medicine. Did I understand Dr. Awad to say that rapamycin was thought to potentially help CCM3 patients specifically?
No, you misunderstood. It's not CCM3. It's a mutation called PIK3CA, PIK3CA3, PIK3CA activation mutation. And that mutation has nothing to do with what you inherit as CCM1, 2, or 3. It's another mutation that develops in the lesion itself and drives the lesion. So it is something that we do not know you have until we take the lesion out. In fact, the only way we discovered this is by looking at lesions that we've resected at surgery. We are developing a blood test for full disclosure that may show in the blood through these microRNAs whether you have the PIK3CA mutation in your lesion. So that is one of the things we're working very diligently on. Just like we're trying to develop the test that tells you, is my microbiome good or bad, which everybody asks me. So I'd love to answer that for you. That's what we're working on.
Okay. All right. So shifting to biomarkers, there's a lot of questions.
All right.
Will the biomarker blood test work for children?
It is not for people under 18 at this time. So the FDA approval process requires us that we either test below 18 or test above 18. And just because of numbers, we went with the above 18. It's not like we don't love the children the same. I think it will be the very next phase to use the same approach. It will go a lot faster after we share what we discovered, because now you don't have to go into the haystack, you know, to look for the needle. Maybe you do that. So Children's Hospital of Boston has been looking at children's urine for some of the same biomarkers that we've studied, VEGF and others, and they see a signal effect, but they really haven't had enough patients bled versus not bled to make it meaningful yet. But I think it will go very rapidly into children once we know that it works in adults.
Good. Okay. I'm curious, if approved, will the biomarker be used to evaluate for possible bleed as an alternative to MRI? Would both tests be used together? Okay.
Yeah, of course. That is a very, very good question. You know, like with every blood test, sometimes people may have the blood test and then undergo a confirmatory MRI. The blood test will not be cheap. So I'm going to start by saying that, because if you have to have relative levels of very uncommonly assayed proteins and so forth, and the company is going to go into this market to develop this, like take it from University of Chicago and develop it. They are going to charge, you know, probably many $100s per pop for the test. So it may not be cheaper than MRI. You know, it may complement MRI in cases where I personally think the MRI is not going to go away. It's going to be the first test to know whether you bled or not. But if you didn't get the MRI in time and the MRI cannot tell if your lesion had bled or not, it will help.
Okay. Is there an estimated time frame for when the biomarker testing might be available outside of a trial?
So I can tell you that we are engaging companies already now to show them under non-disclosure agreements what we're discovering so far, to try to recruit a company that's gonna develop this relatively early, start early in their partnership to develop it. That may or may not happen. You know, companies may want to wait till we complete the testing and then they take it, you know. So we are trying to get it to companies now, but I think the earliest that it could become a blood test is within 5 years.
Okay. You mentioned that mRNA as one of the biomarkers. Is there any confounding with people who have received the mRNA COVID vaccines?
Yeah, so it's not mRNA, it's microRNA, it's miRNA. So for the biomarkers, we study what we call microRNA, miRNA. It has nothing to do with the messenger RNA. So the mRNA of COVID vaccines is a messenger RNA that has to do with the COVID virus. We have nothing to do with that. These are fragments of RNA that run in your blood that reflect the genes in the brain. They have nothing to do with COVID. They will not be affected by COVID or confused. So microRNA is in the blood test. mRNA is what's in your vaccine. One is called micro, the other is called messenger.
Okay. Do you think the insurance companies would cover the cost of this, or is it way too early to know that?
I think the way insurance, the first answer insurance companies will tell you is no. They always do. They did that for genetic testing. They did that for everything. But the way we turn it around is we are the community. They are not. They work for us, and the doctors and the patients are going to be the ones that drive this. So if the patients think it is important to have this test, and the doctors show that it is scientifically valid based on level 1 evidence in our guidelines, then the insurance companies will be forced, happy or unhappy, to pay for it. So that's my mission, is to get it there. You know, I don't care what they think. We, you know, if we have a good thing for the patients, they better pay for it. You know, but before that, we have to show that it's good, right? And that's what all this kind of tedious science is all about.
Okay. All right. I'm going to switch to a COVID question. What about COVID boosters at this point? Do you think that people with CCM should be getting a 4th COVID vaccination, so a second booster?
So let me tell you what is known now about the 4th dose, and it is a changing landscape because the disease is changing. So what we initially developed the vaccine for was for the very severe illness that people who are not vaccinated go into the hospital for. And this happened in about 6% of people who caught COVID the first time. With the Omicron, that rate was still high, but it's not as high as 6%. It's more like 2.5%. But still, Omicron affected so many more people that the hospitals filled up again with unvaccinated folks. But the vaccinated people who got vaccinated after the first wave did not get into the hospitals by and large. So I give you an example. At the University of Chicago, in the Omicron period, the last few weeks, we had a time when we had half of our beds filled with COVID, and there was not a single one who was fully vaccinated. Okay, so they were either, oh, I received the shot a year ago, or I never had the shot. So the ones who received the 2 doses and then the booster had varying amounts of cold symptoms, but rarely had to be in the hospital and never were on a breathing machine and so forth. So that's what we know. Now, the effect of the booster wanes after about 6 months. So those of you who really got it early, like me, are now at that point where we worry, is our booster covering us? The problem now is that the transmission in the community isn't that high anymore. So after this big wave of Omicron, at least in many parts of the country, I don't want to speak for all the parts, because there are parts that have very low vaccination rates where the big wave hasn't hit yet. You know, but you know, if you live in cities where the vaccination rates are high, you know, the disease isn't theirs anymore. So the question, and people are removing their masks and starting to go to restaurants and so forth. So the question is, will we have an uptick as that 6-month people start to wane off, or is the pandemic dying? And this is what the community is trying to answer. And the answer will happen probably in the next month or so, because remember, people started to receive the booster last September, mid-September. So therefore, you know, very few people have had it for 6 months. So we don't know. I may be one of those that will get it, you know, because mine is waning. So we have to wait and see. I think, is there a downside to having a 4th shot? First of all, right now you have to basically lie to get one. Yeah, that's not allowed to be given. So you can walk into CVS and not tell them that you had another one and get one. But the problem there is that we don't know if the immune response that you get, some people have some symptoms from the vaccine and so forth, whether that's justified with the illness waning. It was justified when the illness was killing people. You know, so I think if you're not vaccinated, go get your vaccine and get the booster. But if you're vaccinated with 3, wait another few weeks till we have the answer on this. The state of Israel, who was leading the rule on, they basically were the ones who did the 3rd dose before anyone else in the world. They are not recommending a 4th one. So, and they were ahead of us. In Israel, people started to get the 3rd dose about 6 months before us.
Okay. All right. Now I'm into random questions. We have a few more
There
minutes.
was a Botox question, right? You want to?
Yeah, I can ask that one.
Yeah.
I'm asking for a friend. Dr. Awad has talked about Botox and patients with CCM should avoid it. Does he have the same opinion regarding facial fillers with products such as Juvederm?
No. So facial fillers, have their own problems. People can get allergies and scars from them, but they do not activate the TL4 pathway the way the Botox toxin does. So what happens with Botox is that the actual toxin that makes the muscle weak goes into the nerves. That's why it's called botulinum toxin. And it actually activates in the brain the signal called TLR4. Now, most people, if they activate their TLR4, there is nothing wrong with them. But if you have a cavernoma, the TLR4 is turned on, and that's what causes cavernomas to grow. So that's why people with cavernomas are vulnerable. I'm not sure everybody who gets a Botox will have a problem, just like people can take cocaine and not get a problem, but another person will have cocaine and bleed from their cavernoma. So the issue here is that we have a mechanism that is very specific to cavernous malformation, and we reported it in a very compelling patient who every time she got her Botox, she had a bleed in her cavernoma. Every single time. And then once she stopped Botox, there has not been any recurrence of bleeds.
Okay. I have some really, really good questions here. If you had a lesion resected that was a bad actor with a PIK3CA mutation causing the bad acting, we think, what about the other lesions that you have? If you have the familial form, are they more likely to have a PIK3CA mutation? Are they more likely to bleed? Or does each one have its own independent?
Yeah, each one has its own independent likelihood of getting a PIK3CA mutation. But we don't know how often PIK3CA mutations just happened in the background of the brain. In fact, there is a scientist whose work is to do this. His name is Chris Walsh in Boston, who looks at autopsy brains and brain tissue of all kinds for these somatic mutations, because we don't know how often they happen. But if they happen in a cavernoma, you're screwed. So that's kind of the problem. Let me tell you another thing about PIK3CA. I think I have a minute to tell you that. There is going to be a paper that's accepted now that's coming out in the next maybe couple of weeks in Nature Cardiovascular Medicine from the Duke group and us, where we did something very, very difficult, which is for the first time, we looked at the developmental venous anomaly next to the cavernous angioma and took a tiny piece of it separate from the cavernous angioma when we were resecting a cavernous angioma. So as I'm doing the surgery and removing your lesion, sometimes I have to separate it from the branches of the developmental venous anomaly, because you all wonder, why is this DVA developed cavernous angioma, etc. So as it turns out, the developmental venous anomaly in every sample that we looked at has a PIK3CA mutation in it. So that's probably what forms a DVA. And then if you get a second hit, you get a cavernoma. So there you have the PIK3CA as a sensitizer, and alone it doesn't do anything. But if you happen to have a CCM mutation next to it, then a CCM will develop. So, and we've shown that in the CCM lesion that has additional mutations than the PIK3CA. So that was very tough work. You know, I would have never imagined would be able to have that type of detection work. And that's because of the excellent work in Dr. Marchuk's lab. We would basically be taking these tiny little things, a pinhead sample, and sending it to them, and it's there, it's real.
Okay.
Sorry about going on that,
Oh,
but
no, no, no,
but
no.
I think people, the next
No,
thing
it's
people are
important.
gonna say, oh, that paper came out, I have PIK3CA.
Yeah, no, I'm really glad that you mentioned it, because it's important, and it's great that we're getting a little preview. A basic question about PIK3CA is, are the mutations diagnosed through a blood test or only through tissue biopsy?
Yeah, right now only through tissue biopsy, but we are hoping that if there is enough of the mutation, like the lesion is big or the venous anomaly is big, that we will detect the microRNA of the PIK3CA in the blood. Okay, so that's what we're kind of working on. It's not easy, and it's gonna be a little while before we can prove it. In this Nature cardiovascular research paper, there were, I think, 13 patients with only DVA, no CCM, and we took the sample from their blood, and we weren't able to detect the microRNA. So we are very encouraged that we'll be able to develop a blood test for the venous anomaly PIK3CA mutation in the blood.
Okay, I have one more question. That's a cool question. And if you can't answer it on here, that's fine. We'll get back to her later. I was negative for the 3 gene tests for cavernomas. UCSF and Duke feel that the 20 or more lesions I have are de novo cavernomas. But what does it mean that I have a new, what does it mean? I read there was a new variant, MAP3K3 variant. Do you know anything about this? And for her, they won't treat her AFib due to the cavernomas because of the blood thinners.
Right, so let me answer the AFib question. I think if the lesions are having bleeding, if they are there and they have not had symptomatic hemorrhage, you are better off treating the AFib with blood thinners. We have enough evidence that it is safe to treat the lesions with blood thinners if you have AFib, because without the blood thinners, you're gonna have a different type of stroke from AFib. However, we are screwed if the lesion is bleeding. If the cavernoma is bleeding, then nobody will use blood thinners, and you shouldn't use blood thinners. Then there are things like Watchman procedures and other things to treat the AFib, so you are not on blood thinners. So that's, I think, more important. It's going to apply to many patients. In terms of why do people have multiple lesions and screen negative? There are many reasons for that, and every patient is a little bit different. Some of them could be radiation exposure that they didn't know about. Some of them could be a mutation we do not know. It's not PIK3CA alone. If you have a PIK3CA in every cell in your body, passing it on to your children, you die from cancer rather than cavernous angioma. So it's just not a thing. So why do you develop multiple lesions? Maybe there is a big venous anomaly across your whole brain. That is a possibility. We have such cases that have big venous anomalies, and then they grow cavernomas on them. And there are patients who have what we call hemorrhagic microangiopathy. They are not actual cavernous angioma, but they look on MRI like cavernous angioma. And unless you've taken a lesion out, you don't know for sure. So I cannot tell what your case is, but it's not easy. And we struggle with every one of these cases. I know that Connie sends me occasional cases for me to look at like that, because they're not easy.
Yeah. And I've been getting actually more since COVID. This is just completely anecdotal. I do a lot of genetic testing, and I have been getting a lot more people who are over the age of 60 showing up with what looks like multiple CCM and testing negative.
Yeah,
And
so over 60, if they are hypertensive, have cardiovascular disease, we usually call them hemorrhagic microangiopathy, and we're working on that disease in parallel to cavernoma.
last question, and then we'll go. I'm just finishing up her question. She mentioned MAP3K. How is that playing a role in CCM? Do we have an understanding at all?
In her CCM?
No, in general.
So what we know is that if you have any familial type of the disease, inhibiting PIK3CA does help stabilize the lesions. This happens in the mice. I don't know that that's true in the humans. So there may be some PIK3CA activity happening in the background, even if your lesion doesn't have a somatic mutation for it. When we look at the lesions in the familial cases, about half of them have PIK3CA mutations.
Is that the same?
There may be more.
Is that the same as MAP3K, or is that different?
So MAP3 is another somatic mutation.
Okay.
So I didn't know if you were asking about MAP3 or PIK3.
MAP3.
Yeah, so MAP3 is another mutation that is commonly seen in cavernous angiomas, somatic mutations that may play a role in their development.
Okay. All right. That's all we need to do tonight. Thank you, everyone. There is a link to the support group in the chat. It's been sitting there for about 10 minutes. Darla says, grab it now, or you won't be able to access it. So please, if you're going to join support group, do it now. And I want to thank you all. This has been wonderful. And thank you for joining us and sharing all of your knowledge and wisdom.
Thank you so much again. And call us if you have a bleed and you want to enroll and help us finish the trial.
Absolutely.
All right. Thank
Okay.
you. Bye.
Bye-bye, everyone.
Bye.